All right, hi everyone. Welcome to topic 2, DNA function and point mutations. Okay. Today we really want to understand the relationship between DNA and proteins. Okay, so remember, what is the job or the function of DNA in general? And that's to provide instructions for building proteins. Okay, so if we change the DNA, then we change the proteins in most cases. Okay, so the proteins in cells and in living things are responsible for a wide range of things. And so if we change those proteins, we're going to have effects. It can be on the appearance of an organism, it can be on the regulation of an organism, it can have effects on how it transports materials, how it breaks materials down with enzymes, how it can do photosynthesis or cellular respiration. Okay, so anytime we're changing those proteins, in many cases, they'll have a change in the cell. So protein synthesis is the process of going from DNA to a protein. Okay? And so the first step of protein synthesis is to make an mRNA copy of the DNA. So this is a complementary copy. So if my DNA is TAC, then my mRNA is going to be complementary to that. So it's complementary to T, A. What's complementary to A? Since RNA does not have thymine, it's going to be uracil. So instead of thymine, mRNA always has U or uracil. Okay? So what's going to be complementary to my A? U. Complementary to my C is G. Okay, and there are mechanisms in the DNA sequences that tell the cell which strand should it read. Should it read this one or this one? Okay, but if I'm ever giving you a test question, I'm just going to give you the one strand that it's reading to make the protein. So after it's done making our mRNA strand, the ribosomes read the mRNA strands into groups of 3. Each group of 3 is called a codon, and the codon tells us what amino acid is going to go next in the sequence. Okay, so the codon AUG, my code AUG is the start codon. So it actually tells the cell this is where the protein starts, and what amino acid does it code for? Methionine. Okay. So you can see here, AUG codes for methionine. AUC codes for, let's see if we can find it in here. Was it IUC? What was my codon? I'm sorry. AUC. AUC codes for isoleucine. Okay, and so on. So each codon, each 3 letters of my mRNA codes for a different amino acid. And we put all of those amino acids together, we get a protein. And based on the sequence of amino acids, my protein will fold in different ways. So some of you asked, well, why does the protein fold differently if you have different amino acids? Well, that's because some amino acids are polar, some amino acids are nonpolar, some amino acids have a negative charge or a positive charge. So, for example, amino acid that's positive is going to be attracted to an amino acid that's negative, and it may actually fold my protein, right? So the different characteristics of the amino acids will cause my protein to fold into different shapes. So if we change the amino acid sequence, then we change the shape. And if we change the shape of a protein, it may affect its function. Maybe it doesn't work as well. Maybe it doesn't work at all. Okay, so if we change the shape, it may still work as well. Okay, if the change isn't too severe. But yes, if it will change the protein shape. So we have 3 different types of point mutations, and these are mutations that just change one amino acid. So substitution is when we change an amino acid to a different letter. So my original sequence was TGG-CAG, and my cytosine here, it got mutated into a thymine. This is my substitution mutation, okay? One nucleotide was substituted for a different type. Here we have insertion, so this was my original DNA sequence, but the letters TAT or thymine, adenine, thymine have been inserted into my DNA sequence, so it's insertion. And deletion, here part of my DNA sequence has been deleted or removed. Okay, so which ones of these have the largest effect on the amino acid chain? Substitution usually affects one amino acid or no amino acids if it's a silent mutation. Insertion and deletion are usually frame shift and they affect all of the amino acids in the sequence. So let's take a look at that. For example, here we have, if we have a substitution mutation, I believe here we have a substitution mutation. One second. Here, this C has been substituted for a G. So how did it change my mRNA sequence? Well, now my mRNA sequence, now this codon is UGG. Well, it was originally UGG. Now it's UCG. So my substitution mutation changed one of my codons, so it changed one of my amino acids. Here we have an insertion mutation, okay? So in this case, the letter A was inserted into my DNA strand. Before it was TCCC. Sorry, that was not my mutation. One second. Okay, so here's my insertion mutation. It looks like my insertion mutation is here. Originally, my strand was GTAG. Now it's GTATG. So this nucleotide here was inserted. So before each of, this was my codon, CAU. My codon was CAG. Uh-oh, my codon is not CAG anymore. It's shifted over. Now it's ACA. Here my codon was UGG. UGG is over here. Uh-oh, I'm shifted over one because of my insertion mutation. So my codons have now shifted over. This is called a frame shift mutation. It happens with insertion and deletion. So these two here are called frame shift. So here we go, a frame shift mutation. It's caused by the deletion, the addition or deletion. So insertion or deletion mutation, okay? And it basically shifted over my codons. Okay? Why do they generally result in a non-functional protein? Because we literally have changed all the amino acids in our protein, so we've completely changed our protein. Okay? All right, let's continue on. Now, a silent mutation, this is when we have a mutation, but it doesn't change the amino acid. So here's my original codon, my original mRNA strand, my mRNA strand formed by the original DNA. And you can see here I have the codon CCU. I have a mutation. So this U is now a C. However, did it change the amino acid that it coded for? No. So sometimes we can have mutations, like if this C changes to an A, it doesn't change my amino acid at all. So sometimes we can have mutations that don't have an effect on the amino acid chain. That's because, for example, if the codon CCU is now CCC, it doesn't change anything. They both code for proline, okay? And if it's changed to CCA, it's not going to make a difference because it still codes for the same amino acid. So these are called silent mutations. And that is the end of our topic 2. You can take a look at these, but we'll talk about them more later. They're just 3 case studies or 3 types of mutations that cause disease and symptoms. And so, I mean, it would be good to look at, for example, here we have cystic fibrosis. We have this mutation here, which is a deletion of the 508th triplet, okay, of the DNA code for the chloride transport protein. And so as a result, the protein cannot regulate chloride ion balance in the cell. And so things like our glands, pancreas, intestinal glands are affected because these are all all of these glands and organs require the transport of chlorine in and out of the cells. Okay, so why do the symptoms make sense? Because they are related to the transport of chlorine in and out of cells. Okay. And we have, what effect did it have on the protein, our mutation? Well, it made it, I think, non-functional, right? So as a result, the protein is non-functional and it can't carry out its function.